All right, well, thanks, Gustavo. It's great to be here and to be able to participate in this challenge exercise. I'm really excited about this. I think as an experimental genomics type, my life in the future is going to be to just generate a data set, toss it out there, and let you guys tell us what it means. So, interesting change in perspective. So I am going to talk about the data that went into our part of the DREAM challenge. It is a set of measurements of drug response on a panel of breast cancer cell lines. And really the rationale for wanting to do this is not because we care about predicting the responses of breast cancer cell lines, but rather we're trying to use this to gain some insights into how it is that we ought to better manage breast cancer. And so we hope that some of the analyses that come out of all of this will inform us on this. And so I've divided this talk into three parts. I want to take just a minute to give you my perception of where we stand on understanding what the sort of the omic parts list in breast cancer really is today, because I think we are at a remarkable point in time in that front. I'm then going to talk a little bit about some of the work that we've done in terms of functionally interrogating that landscape, both experimental and computational models. And we didn't get to participate in the challenge, so I'm going to show you what we did, but who knows how we'll stack up. We'll find out. And then the last part of this, I'm going to talk a little bit about where we're going in the future with the idea of perhaps setting the stage for some future DREAM challenge. So in terms of defining the parts list that is going on in breast cancer, essentially what we're trying to do is to identify the ensemble of omic abnormalities that contribute to the deregulation of a whole variety of what Hanahan and Weinberg have come to call cancer hallmarks. So it's not just proliferation, it's not just apoptosis, it is angiogenesis, it is energetics, it is genome instability, and so on. And so I think that one of the things that we are appreciating is that what's going on in omics is the deregulation of all of that. And so what we're trying to do is to really discover what those driver abnormalities are that enable these cancer hallmarks. And so essentially what we're trying to do in breast cancer management in general is three things: to try to interpret all of these genomic abnormalities in terms of how it is that they deregulate some of these key circuits. Having known that, then the idea is to try to develop therapeutic strategies that either reverse the effects of these aberrations or kill the cells that carry them. And as we go down the road, I think one of the things that we increasingly appreciate is that these circuits that we're trying to attack are reasonably plastic, and they evolve mechanisms of resistance pretty rapidly. And so we also need to begin to get at what those mechanisms are. So in breast cancer, as I say, we are at, I think, a remarkable point in time, and that is we've been in the genome community for the last decade trying to figure out what was going on in breast cancers. And I think this year, in publications that appeared in June and September in Nature, we've pretty much completely defined the genomic landscape of most human breast cancers. These publications represent analyses of on order 3,000 or 4,000 breast cancers of a variety of types. And so I think that we know pretty much what's going on as a result of those complications. And I'm going to go through this very quickly. I think that you all appreciate it. But one of the things that comes out of this is that there are a variety of genomic and epigenomic mechanisms that contribute to the deregulation of the cells and that drive cancer pathophysiology. And essentially, every abnormality that you can think of comes into play in one or more of the cancers that have been analyzed. So that's daunting. Another thing that's daunting about it is that a typical tumor, based on these studies, will carry a few relatively well understood so-called driver abnormalities and literally tens to hundreds to thousands of poorly understood abnormalities, some fraction of which may contribute to pathophysiology of cancer. And I think that just exactly what constitutes a driver and a passenger abnormality is far from well established at this point. And I think that one of the things that we're going to appreciate as we go farther and farther down this road is that what we think of passenger genes today may in fact become driver genes tomorrow as we understand how it is that they contribute to cancer. But based on what we know so far, just summarizing all of those publications, there's about 45 regions of recurrent amplification or copy number abnormality, about 10 different subtypes of cancers that have been defined. Spectrum of abnormalities differs according to subtype. Pretty high mutation rates, so that there are a lot of somatic abnormalities in these cancers. There are a few recurrent mutations and many rare ones. The recurrent ones are anywhere from 80% to 50%, and then when you get down in here, you're 1 or 2% or one-offs. So understanding this is somewhat challenging. And as you start thinking about the emergence of resistance, one of the things that is a little bit depressing is that there seems to be really relatively few smoking gun drivers of this, and sort of an ensemble of different ways that one can get to resistance. And so that's something that we're going to have to deal with. But what it does now do is to give us a collection of abnormalities that we can think about beginning to associate either with outcome or therapeutic response. And in fact, two of the challenges that we have under discussion in this meeting address these issues. And so that's the state of it. What I'd like to do now is to talk about what we do with this information and what we're trying to do with it. And really, how do we functionally interrogate this landscape? And from our perspective, the reason that we care about functionally interrogating the landscape is because we believe that the genomic landscape will determine how it is that the cancers respond to the growing number of therapeutics that are now being developed. If you look in the pharmaceutical industry, there are going on 1,000 compounds that are somewhere in clinical testing. And one of the things that we need is some sort of, I think now, computational or theoretical understanding of how it is that we can identify which ones of these abnormalities will best predict response to these targeted therapeutics. So how do we do that? And I think that it is true that it's not going to be possible logistically to analyze these drugs in enough breast cancer patients to be able to establish those kinds of associations just using clinical material alone. So we need biological models to do that. And the one that we've been developing is this collection of about 60 breast cancer cell lines. And the idea is that the molecular diversity of these cell lines is rich enough that it captures a lot, but not all, of the molecular diversity that are found in the tumors that we're trying to treat. And so we spent a lot of time trying to understand what the omic features of these cell lines are and how it is that this information compares to what it is that has been generated now in the TCGA and ICGC and other projects on primary tumors. And so we've looked at RNA, protein, methylation, we in collaboration with the community. And by and large, in most of the omics space, the match between what we measure in the cell lines and what gets measured in primary tumors is pretty good. It's not great, but it's pretty good. Probably the biggest difference between what we see in cell lines and what happens clinically is in the area of epigenomics. I think that the epigenome is reasonably plastic, and when you grow cells on plastic, you do get some changes. And so clearly, at least in terms of promoter methylation, the cell lines are more heavily methylated than they are in primary tumors. But otherwise, from a genomic point of view, we see a lot of what's going on in the tumors. So that's good news. And so we think that there is enough of the biology and genomics of primary tumors captured in the cell lines that it is worth trying to use these cell lines as a model system to test all of these therapeutic compounds in. And so we've been doing this for a while. The idea here is that we grow the cells, treat them for 72 hours. We treat them in 9 different drug concentrations. 3 replicates and measure the concentration of drug needed to inhibit growth by 50%. And a subset of these data is what has been released as part of the DREAM challenge set that some of you have been analyzing. And what we and you have been doing here is to try to take these data, take the corresponding molecular features of the cell lines that were responding, and try to develop computational strategies to predict drug response. And the reason that we want to do this is because we think that we want to carry these predictors into the clinic with the idea that it will help us to guide drugs into the clinic. So we have been taking several approaches to this. One of them has really been developed by Annalene Damon and Obie Griffith. Annalene's now at Genentech, and Obie is at WashU. But basically, these were support vector and random forest approaches, and essentially it just took the data, it dichotomized them into non-responders and responders, built a predictive signature. There was a lot of leave a few out cross-validation studies done, and ultimately this ended up developing predictors for which we could generate ROC curves that will give us some assessment of what the quality of the predictor is. And one of the things that I'm sure that those of you who have analyzed these data will appreciate, the power to predict response depends a lot on which drug you're talking about. And at least in our own hands, what we find is that we, even with having access to the full data set, we're unable to develop very good predictors for maybe about half of them. And we're able to predict pretty good predictors for about half of them. So, amongst other things, the dynamic range of the data is going to determine to some extent how well you can predict with them. But this is reality. Obie and Annalene also looked at the extent to which the different data types that we have available were involved in the most important predictive features, and nothing in our hands seemed to emerge as the clear winner. In all cases, there seemed to be a few features that were important in all of these different predictors that we developed. So, no winner here. If you threw out the RPPA data, I would have to say that if there was a winner, it would probably be RNA-seq, but not strongly biased in that direction. So, one of the things that we are eventually moving toward is to try to be able to use this information to select treatment for individual patients. And so one of the things that was done in this study was to take all of the predictors that we generated for our best performing compounds, the ones where we had really good high quality predictors, we thought, and we applied those to the TCGA cancer data set. Not that we have outcome information on that data set, but at least we know what the omic features are, and we basically just predicted drug response patterns to all of these compounds that are located here. And there's a couple of messages that come out of this. One of them is that it really does demonstrate that a lot of the power of the predictors comes from the transcriptional subtype of the signature. But the other thing that's interesting is that if you just took this as a representative set of breast cancer patients, if you took a portfolio of 22 compounds, we would predict that we would be able to find one compound that would be really highly effective for every patient. And so it does suggest that this is moving us in the direction of being able to do precision medicine. Let us identify the omic features of a cancer patient, run our predictors, and pick the drug that would have the highest probability of being effective in that cancer patient. And so Obie and Annalene have done that. They've actually developed a individual patient drug selector algorithm and applied to this. And so this is now available, and we will ultimately be going forward thinking about how to test this clinically. I think one of the things that we can think about collectively is that there's a certain caution here, and that is that high relative sensitivity in vitro, which is what we're using as the basis for this predictor, doesn't necessarily translate into clinical response. It could be that there is a subset that are highly effective in vitro and that none of them are highly effective in vivo. So we have yet to have a good idea as to how to make the in vitro to clinical translation, and that's one of the things that we want to work on in the future. Well, that's one approach to interpreting these data. But there may be, this particular approach may be problematic. So the reason that it may be problematic, I'm going to return to this slide that I showed before. The problem here is that these key regulatory pathways that we think are being deregulated and driving therapeutic response can be deregulated in many different ways as you go from tumor to tumor. So one tumor might be deregulated as shown by the X's. In this example, the next tumor might look like this, and the next one like this, and the next one like that. And so if you tried to analyze that just using sort of straight-up machine learning strategies, you may not get anywhere because the patterns are all different. There's no single predictor that would allow you to infer a response. But if you map these things onto pathways and then interpreted this in the context of pathway deregulation, I think that this might be a productive way forward. And so Laura Heiser, who you're going to hear from momentarily, has been working with Josh Stewart and David Hausler and the colleagues at Santa Cruz to apply their program, this paradigm program, that does exactly that. It takes the diverse data sets and integrates them to calculate pathway activity levels for a whole variety of literature-curated pathways. Greater than 1,300 curated pathways have been analyzed in this way for all of the drugs that we've been talking about. Now, the problem with this is that this actually requires that we know which of these low-frequency aberrations are actually functional. And I don't think that we know that right now, and I'll tell you a little bit about how we're going about trying to deal with that. But this is what it is that we're trying to do. And so Laura's done this. What I'm showing you here is data on a mixture of tumors and cancer cell lines. These are now pathway activity levels, not gene expression levels. I'm not going to go into the details of all of this, but basically what these analyses show is that the tumor and the cell lines show reasonably similar pathway activity signatures, and also, whether you're talking about cell lines or tumors, the activities show strong subtype specificity. So that suggests to us that the cell lines, again, are capturing the pathway activity that we think we need to interpret drug response. And so what we want to do is to move forward in terms of how to use this. The problem is that it's a little daunting to think about dealing with the 1,400 pathways. And so Josh and colleagues have eliminated the pathway redundancy, producing giant hairballs that now allow us to compare sensitive and resistant populations and identify within these large networks nodes or subnodes that are either upregulated or downregulated differentially between the comparative populations. And so this is how it is that we are thinking about interpreting these data. These things could be associated with subtype, or they could be associated with drug sensitivity or resistance. And so the problem here is that we, right now, don't know what most of these nodes do. And so one of the challenges for the future is to figure out what aspect of cell biology these things are influencing when they are coordinately deregulated. And so sort of thinking forward for the future of the kinds of data that might be interesting to interpret in this context, one of the things that we've done is to develop an experimental enterprise that will allow us to, we think, efficiently interrogate each one of these networks and to understand how to map that onto the cancer hallmarks that we care about. And the way this is done takes advantage of some technology that was developed in Finland by Juha Rantala and colleagues, now transplanted to Oregon. And basically, what's going on here is that we make microarrays of tens of thousands of small spots, each of which contains an siRNA that can be chosen to target some of these pathways that we want to interrogate. And each of these spots will host on order 100 cells, and these things are read out using an image analysis system that looks at anything that one can develop using some sort of an immunohistochemical assay. And so, for example, you could look at EDU incorporation, p53 binding protein 1, expression levels, and so on, and quantify how it is that these things change as cells grow on these spots and get autotransfected with the siRNAs. And so the nice thing about this particular technology for this network node analysis is that these are pretty easy to build, and so we can build one for every node. And so it allows us to efficiently go through that. And what Juha's now introduced into the laboratory are a whole variety of immunohistochemical assays that have been developed to try to come up with surrogates for all of the cancer hallmarks that we care about. And so we can interrogate things that are involved in proliferation. We can look at EDU incorporation. We can have markers for differentiation. We have markers that look for DNA repair, apoptosis, et cetera. And so we think that we have enough assays at this point that we can begin to quantify most of the cancer hallmarks that these networks and genes are influencing. I won't go into this in detail because we're running out of time here, but these can be quantified using image analysis. And so what have we done with this? And basically, what we've done is to start down the road of interrogating the things that we care about in terms of this network function. So one of the things that we wondered is, okay, we've got all of these low-frequency abnormalities. Do these things actually function? And so working with Juha and Ray Cho and Eric Collison and Laura, basically what we've done is to identify 450 genome aberrations in TCGA, found instances of the cell lines in which those were aberrant, and now we've interrogated all of those with siRNAs. And we can ask the question, if you look at genes that are amplified, and knock them down, some of the responses will be that the cells grow faster. You knock them down, some of them, you'll get inhibition of growth in cells that carry the aberrations. And if you just look at some of the things that you would expect to show up in this, MYC, cyclin E, et cetera, these are things that we know about, so we think that the system is telling us the truth on that. And so, there are about 20 new genes in here that we think are probably driver genes that are relatively low frequency, but we think are important. And you can do the same thing with these low frequency mutated genes. In this case, you would expect if they are tumor suppressor genes that you would get higher knockdown efficiency in cells that are wild type at the target. P53 shows up, PIK3CA2B shows up, things that we would expect to show up in that category. So again, it seems to be telling us the truth, and so I think that we're picking up gain of function and loss of function mutations there. So it's now beginning to teach us which ones of these abnormalities we think we could use to help us build some of those networks that we've been working on before. So the next thing that Laura's done is to go in and start challenging these networks. One of the things that one would hope is that if you see a whole bunch of genes that are coordinately upregulated, when you knock them down, you would see the same response independent of where within that network you knock down if they're all working together. This happens to be a FOXM1 hub that's upregulated in basal cell lines. When you start knocking the genes down in that, we see a pretty concordant set of responses for all of the genes in the network. So that's sort of consistent with what's going on. And as you might expect, if you attack cells that have high pathway activity levels in this network with drugs that attack into this actually DNA repair network or into this mitotic apparatus network, rather what you find is that they respond preferentially well. So that's interesting. That's as we would have expected. Here's something that I didn't expect. So this is relatively new data. Essentially what Juha's done here is to take a slightly different approach to network development, a sort of string-curated literature and protein-associated networks. We started knocking them, well, first off, we formed the networks and then started knocking them down with siRNAs, as I've described. And one of the interesting things is that you get, again, coordinate behavior when you start knocking these networks down. So these genes coordinately respond to siRNA one way. These respond a different way. So, interestingly enough, when you knock these down, it increases proliferation. And if you take those genes then and look at the expression levels of those in clinical data sets, one of the things that you find is that higher expression for these seems to be associated with better outcome. These, on the other hand, when you knock them down, it decreases proliferation, suggesting that these are really driver genes. And interestingly enough here, high expression of these things is associated with worse outcome in the primary tumors. So, again, now what we're beginning to be able to do is to map functional information from the networks onto the behavior that you see clinically. And I think that that's going to be particularly powerful in the future. So there's a lot more that's going on here. We're not talking about it yet, but one of the things that does happen when you start knocking some of these genes down is that you get profound changes in things like differentiation status that we think are going to have a major impact on how cells are responding to therapy. But that's a story for another day. The last little bit that I want to tell you about, and something that we need to be thinking about in terms of translating this information to the clinic, is that so far I've been talking, and I think most of us focus on tumor intrinsic biology, what happens within the tumor. But of course we know from the work of many people that tumors do not sit by themselves in the body. They are embedded in a very complex microenvironment. And if you're thinking about metastatic cancer, one of the things that's true is that those microenvironments differ as you go from point to point in the body. And so, you've got cancer-associated fibroblasts, you've got infiltrating immune cells, you've got angiogenic vascular cells. All of these cells are actually contributing cytokines or directly interacting proteins that modify how it is that these networks that we are attacking therapeutically actually work. And so the question is, how do we study this? And back to this microarray format that I've been telling you about, now what we're able to do is, instead of putting siRNAs in these little growth pads that we're growing the cells on, now we put proteins from the microenvironment. And you can do this singly or in combination, and then grow the cells on that and ask, how do the cells that are grown on these different microenvironments respond to treatment? And it turns out that there are a lot of different microenvironments that have already been reported in the literature as being important in this particular process. And so some of them come from all of the different types of cells that tend to invade these cancers. And so what we're doing is trying to make arrays that sort of capture these things singly and in combination. And we're ending up interrogating several thousand different microenvironments for every cell line and every drug that we're testing at this point. And so this is generating a rather complex data set that I hope to bring to this group sometime in the future for your attention, because one of the things that we're going to have to do is to understand in some detail how it is that all of these different perturbations, if you will, on pathway activity influence therapeutic response. But just to give you a little snapshot of some data that was generated by Mark Labarge and Chun-Han Lin in his lab at Berkeley a little while back, just shows you what's going on here. These are responses to ERBB2 amplified breast cancers that were grown on pairwise combinations of different microenvironments. And what these little squares here show you is whether or not the cells were either sensitive or resistant to HER2-targeted agents, in this particular case, lapatinib. And I won't go into the detail, but the bottom line is there are a lot of red spots and a lot of blue spots, and it just makes the case that the microenvironments are substantially and quantitatively influencing how it is that these networks are behaving. So I think that we're going to have to begin to bring these data together with the tumor intrinsic information if we're really going to come up with predictors that will respond or that will be effective in the clinic. And so so that's what we want to do. And one of the problems that we have in this field is that it's really easy to generate the cell line data. And so we generate all of these predictors. And then you have to ask, do they work clinically? Okay, well, if you go to the literature, you will find no data to support any of the drugs that I have been talking about and that you've been analyzing. So One of the things I'm excited about is ISPY 2. So this is a clinical trials program that Laura Esserman in particular has spent going on a decade organizing. And it is a neoadjuvant clinical trial. So the drugs are given to breast cancer patients before surgery. And the interesting thing about this trial is that the patients are biopsied before treatment, during treatment, after treatment. Tissue is taken at the time of surgery, and they are monitored for response using MRI. And this is a trial that's now going on in about 20 institutions around the country. So it accrues rapidly, and this is all being organized through the NIH Foundation. And so there are like 10 compounds that are now queued up to be run through this clinical trial. And we have arranged, actually the NIH Foundation has arranged for us to get all of these compounds into the laboratory. And so we are going to be generating the same kind of data that I just talked about for all of the compounds that are going into this trial. And we will then be able to test the in vitro predictors with what actually happens clinically. I think that's critically important. And so, you know, if it works, then out of this comes both drugs and markers that can graduate for future testing. And if it doesn't work, then we are at least in a position to figure out why it is that our models aren't telling us what we want them to tell us clinically. And so then you can go back, we can change the models, we can change the way that we analyze them, turn the crank again, and keep going until we develop the models to the point where they are actually clinically effective. And I think that's where we all want to be with this at the end of the day. So that's where we're going. Now, just relative to the second challenge that we're going to have here, I'll show you one final slide and then I'll be done. And that is the following. You will notice in this trial that they're actually getting the drugs in combination with paclitaxel. And so we wanted to model this properly, and so Jim Korkla has been generating a lot of these data. Analyzed this where paclitaxel was given together, in this case with an AKT inhibitor alone, an AKT inhibitor alone, or we gave paclitaxel first and then AKT, or then we gave AKT and then paclitaxel. And one of the things you immediately see is that schedule is really important here. If you give an AKT inhibitor before paclitaxel, you can substantially diminish the efficacy of paclitaxel, at least in the cell lines. Now, this is not new to us, but I think that this just illustrates the fact that another of the challenges for the future here is to think about not only giving drugs in combination, but what schedule should we be giving them to get maximum efficacy. So that was the story I wanted to tell you. Basically, it's a very exciting time in breast cancer in that the genomic landscapes, I think, are now pretty much established. I think that we and many others in the community are now beginning to develop some pretty powerful model systems that will allow us to begin to integrate these data in ways that allow us to predict therapeutic response. And I think as we go forward, we have the power to think about bringing in the extrinsic influences as well as the intrinsic influences in terms of making these predictors. And how we actually do that is computationally pretty challenging. So these are the people that have participated in what I talked about in terms of the pathway biology. Laura, Obi, Annaline, and Josh have been involved with this. I really need to give major credit to Jim Korkla and Nick Wang, who generated the data that has been part of this challenge, and that was a real multi-year effort. Talked about Juha, and Paul Spellman has led our genome biology effort. So with that, I thank you for your attention, and I hope that some of you will pay attention to the subliminal message up in the upper right-hand corner here. Thank you. Thank you. No.
I don't think, oh yes, it's working. So you've shown how all these microenvironments change the response, but of course the question is your first part of the talk might be only relevant to a certain microenvironment, and you don't know the microenvironment that corresponds to the clinical situation. So I know that, so in your ISPY trial, are you also testing microenvironments to see which one will predict the clinical situation?
Well, it's an interesting point you make. The question is, how do you actually do that? So you're right. We're going to find a whole bunch of different microenvironments that influence a response. The hope is that these are going to computationally integrate into a few pathways that we are deregulating. So, you know, there's a variety of ways that one can deregulate the pathway. One hopes that we can begin to unify that information much as we're trying to unify the tumor intrinsic information so that it gets down to relatively few pathways. If we can do that, then what we will do is come up with immunohistochemical technologies, you know, markers that will allow us to interrogate those same pathway activities, sort of intrinsic pathway activities in the ISPY samples. So there are samples are available for that kind of study. We are pointing toward that. It's not, you know, that's probably 3 years from now once we've analyzed those data a little bit so that we know what to interrogate. But we're definitely going there. I think that we have to understand that. But the hope is that if we can understand it, then we can come up with inhibitors that will block the influence of the resistive microenvironments.
One last question.
Yeah. So in a little bit the same direction, but I think one of the aspects that has been coming up is metabolism, cell metabolism. So how do you put that in this context? I mean, because metabolism, of course, has to do with the microenvironment because nutrients comes from outside, but somehow there's a homeostasis of the cell, so pathways are also implicated. So that seems to be like a point of convergence of these two things.
Right. So this is actually, I mean, what I'm showing you here is a small piece of our integrative cancer biology project, which is really taking a broader look than I've described here at these networks that we're interacting with. And one of the things that is featured as part of that is sort of the interaction between the AKT part of this network and the whole metabolism bit. And so we have a sub-project that's actually aimed at trying to bring that in. I mean, I have to say that you know, bringing in the metabolome is daunting. Not that tumor genomics isn't daunting, but that's even more daunting. And so it's a little less developed, but we're trying to go there. It is already apparent, though, that the sort of the basal metabolism, the metabolic pathways usage varies according to subtypes. So we already know that, and we're working to try to bring it in. And we actually have the data, and we're doing a fair amount of mass spec on this with Trent Northern to get at it, but it's in a primitive stage of analysis.
Thank you.
Okay, let's thank Joe one more time.